The theory of molecular genetics provides the framework for understanding how genes function at the most fundamental level, describing the structure, replication, expression, and regulation of genetic material. It is the cornerstone of modern biology and has transformed medicine, biotechnology, and our understanding of life itself. At its core, molecular genetics explains how DNA, the molecule of heredity, stores instructions for building proteins, how those instructions are copied and passed to new cells, and how genetic activity is controlled to ensure proper development and function. The foundation of molecular genetics was laid in the mid-20th century. The discovery of the double helix structure of DNA by James Watson and Francis Crick in 1953, building on Rosalind Franklin's X-ray diffraction images and earlier research by Maurice Wilkins, revealed how genetic information is stored and duplicated. DNA consists of two strands of nucleotides paired in a complementary fashion, adenine with thymine, cytosine with guanine, providing a stable mechanism for both storing instructions and copying them during cell division. This discovery set the stage for understanding the processes of replication, transcription, and translation. DNA replication ensures that when cells divide, each daughter cell inherits an exact copy of genetic material. The double helix unwinds, and specialized enzymes like DNA polymerase use each strand as a template to build a new complementary strand. This process is remarkably accurate but allows for occasional mutations, which contribute to genetic diversity and evolution. Transcription is the process by which DNA's instructions are converted into messenger RNA, mRNA. Here, RNA polymerase reads the DNA sequence of a gene and produces a single-stranded RNA copy. Unlike DNA, RNA uses uracil instead of thymine, but it carries the same coded information needed to build proteins. Translation follows, taking place in ribosomes, the cellular factories. The mRNA sequence is read in sets of 3 nucleotides, called codons, each specifying a particular amino acid. Transfer RNA, tRNA molecules bring amino acids to the ribosome, where they are linked in the correct sequence to form a protein. This flow of information from DNA to RNA to protein is known as the central dogma of molecular biology, first articulated by Francis Crick. Proteins, in turn, carry out nearly all the structural, enzymatic, and regulatory roles essential for life. Equally important is gene regulation, the ability of cells to control when and how genes are expressed. Not all genes are active in every cell at all times. Complex networks of regulatory proteins, enhancers, silencers, and epigenetic modifications like DNA methylation ensure that genes switch on or off in response to developmental cues and environmental signals. This regulation allows for cellular specialization, enabling a single genome to produce the diversity of cell types in multicellular organisms. The theory of molecular genetics has advanced beyond basic replication and expression to include powerful insights into genetic engineering, genomics, and biotechnology. Techniques such as DNA sequencing, PCR, polymerase chain reaction, and CRISPR-Cas9 genome editing have revolutionized research, medicine, and agriculture. Scientists can now read entire genomes, diagnose genetic disorders, and even edit specific genes with precision. Molecular genetics continues to evolve, integrating with systems biology and epigenetics to provide a more complete picture of how genes interact with each other and with the environment. From personalized medicine to synthetic biology, the applications of this theory are reshaping the future of science and technology. Ultimately, the theory of molecular genetics answers one of biology's most profound questions. How does life replicate, sustain itself, and adapt? By explaining the molecular processes behind heredity and gene expression, it has not only illuminated the nature of life, but also opened new horizons in science, health, and human understanding.